continuing on with our look at how microbes are able to cause disease, we will take some time to explore virulence factors. Virulence factors are the advantages that pathogens have that really make them able to cause disease and then determine the extent and severity of the disease. The virulence factors are going to come from genes that the pathogen is going to have, either in its chromosome or from a plasmid that it has picked up, somewhere along the way. When the genes that encode for virulence factors are inactivated, then their virulence is diminished, and in some cases they may not even be able to cause disease. So what we'll look at in this section are the virulence factors of bacterial and viral pathogens, and in the next section we'll get to the virulence factors of eukaryotic organisms. So the first thing we look at are the virulence factors for adhesion. So adhesion allows the organism to be able to stick to a particular surface or a particular cell in the body. And we find different types of adhesins, which are the proteins that are responsible for this, in bacteria, viruses, fungi, and protozoans. So we see different proteins involved really in all groups of our pathogens except for the parasitic worms. So in this table you can see a few of these different types of adhesins and the bacteria that have them and where they're allowed to attach. You'll notice that the attachment site is pretty specific, and that goes back to the idea of the protein having to fit into its exact receptor. So like cholera, for example, which is caused by Vibrio cholerae, can't infect the cells of the respiratory system because its particular adhesin does not fit to those cells. So it's going to attack the epithelial cells within the lining of the intestine because that's where its adhesin protein fits. After an individual has been exposed and after adhesion has taken place, so the pathogen has been able to stick to its surface, the next step is invasion. And this can involve a lot of different mechanisms, but two of the more common ones that we see are through the use of enzymes and toxins. So one way that pathogens can achieve invasion is to get into the bloodstream. So they make their way, through the tissues, get into the bloodstream, and then they can be disseminated anywhere in the body because, of course, we know that we have capillaries in all areas of the body. The downside for the pathogen for this mechanism is that within the blood itself, we have a lot of components of the immune system. So we have different chemicals, and we have antibodies, and we have complement, which is a special type of protein that helps activate the immune system. So the pathogen may not find itself in a very hospitable environment and may be killed or inactivated before it can make its way to another place in the body to cause disease. Now, when we have the presence of bacteria in the blood, that is called bacteremia. Bacteremia involving the pathogens, which are those pathogens that can form pus, is called pyemia. We have viruses in the blood, that's viremia. Toxemia is when toxins are found in the blood, and if bacteria are both present and multiplying, so in other words, they are kind of establishing a foothold, the immune system isn't really taking care of them, then we call that septicemia. So individuals that have septicemia, so they have an active infection of bacteria multiplying in the blood, are described as septic, and this can lead to shock, which is when the blood pressure drops. When the blood pressure drops, this prevents oxygenation to parts of the body. Some bacteria cause shock through the release of toxins. Some cause septicemia and toxemia when they get into phagocytes. So when our phagocytes, cells of the immune system, ingest them, they can trigger inflammation and fever. If shock continues, it can be fatal, because if the blood is not circulating through the body properly and getting to all the organs, then this leads to multi-organ failure, and if it's not quickly corrected, then it can cause death. So we're going to look at a couple of different types of these enzymes and toxins. So the first type that we have are called exo enzymes. Exo here represents extracellular. So these enzymes are secreted by the bacteria, and it allows the bacteria to then invade the host cells and basically get deeper into the tissues. Some classes of exoenzymes are pretty general. You can see these listed here in this table 15.8. Each of these functions in a specific type of tissue to facilitate growth of the pathogen or, to defend against the immune system. So one of these big classes are the Glycohydrolases. And one particular example you see there is Hyaluronidase. Hyaluronic acid is the glue that essentially holds the cells together, especially if we look at the cells of the skin. And if a bacterium secretes some type of hyaluronidase, it dissolves that glue holding the cells together, which makes passageways for the bacteria to then invade deeper tissues. You see collagenase there. Collagenase degrades collagen. Collagen is found in connective tissue, and it's what really provides strength to our connective tissue. And we'll have different types of connective tissue that have different amounts of collagen. But if you have a very rich area of collagen and the bacterium is starting to break that, secrete this enzyme that breaks that down, it again just allows them to burrow and dig deeper into the tissues. So exoenzymes allow the bacterium to come in and, essentially evade the immune system, as well as get deeper and deeper into the tissues that allow them to move away from their specific portal of entry, or to become disseminated away from their portal of entry. So here is this example of, hyaluronidase, where here's the glue, and now that those cells aren't attached to each other anymore, those bacteria can just get right in through there, and this allows them to get to all of these tissue layers that are located below. And then they'll continue to secrete this hyaluronidase enzyme that allows them to just keep going, deeper and deeper. And then here's the example with the collagenase. You've got some connective tissue here. They're showing the leg. And in this example, they're looking at the endothelial cells here, essentially the cells that line the capillaries. And collagen is found in those blood vessels. So if that happens, then the bacterium can actually get into the lumen of the blood vessel, into the bloodstream, and then become disseminated that way. And Clostridium perfringens, which causes gas gangrene, can do that. When we look at toxins, toxin means poison. And so when we consider the word toxin, we think these are poisons to the body. These are going to be substances that are produced by bacteria that allow them to invade and cause damage to tissues. And the ability of these bacteria or other pathogens to produce toxins is called toxogenicity. When we look at toxins, we can have endotoxins or exotoxins. Endotoxins are essentially a part of the organism itself. And the best example that we have of this is that lipopolysaccharide that we see on gram-negative bacteria. Remember when we talked about gram-negative bacteria. We said that, in addition to that thin layer of peptidoglycan, they had this outer membrane that contained a special lipopolysaccharide. It has this lipid A portion here, this core glycolipid, and then this O antigen, which is a polysaccharide. The lipid A is the toxic part of this lipopolysaccharide. During infection, the gram-negatives will release this endotoxin, either when the cell dies, as the membrane disintegrates, or when it undergoes binary fission, so when it's replicating or when it's dying. Lipid A is pretty similar regardless of the type of pathogen, because these lipid A molecules are about the same. They're nearly identical across all of our gram-negatives. So lipid A triggers essentially the same symptoms, leading really to an excessive inflammatory response. The lipid A really just kind of turns our body against us, and it can lead to shock and death in severe cases. Exotoxins, on the other hand, are actually protein molecules, not lipids, that are produced by the pathogenic bacteria. Some gram-negatives are capable of producing exotoxins, but the majority of these are going to come from gram-positives. Exotoxins are very specific in what they do. They're not like the endotoxin. They're not like that lipid A that's pretty general and just launches an inflammatory immune response. The exotoxins are very specific in the reactions that they cause and what type of cells that they interact with. Endotoxin remains stable at high temperatures and requires a lot of heat for a long time. But the exotoxins, since they are proteins and not lipids, can be denatured at a little bit lower temperatures. Endotoxins typically take a larger concentration to cause damage, but exotoxins do not, and that's what's referred to here as our LD50, which is the lethal dose. And so you see here it's very low for a lot of our exotoxins. Botulinum toxin, for example, has an LD50 of 0.000001 milligram per kilogram. So that's a tiny, tiny amount that can cause death. So exotoxins are grouped into categories depending on their target. So we can have intracellular targeting toxins, those that disrupt membranes, or those that we refer to as superantigens. These superantigens are the ones that are more similar to the endotoxins in that it stimulates the immune system and launches this massive inflammatory response. If we look at our intracellular targeting toxins, these are going to target something inside the cells, inhibiting neurotransmitter release or inhibiting protein synthesis. Membrane disrupting toxins are going to do something to the cell membrane, either degrading it or poking holes in it, somehow killing the cells. And so you see we have quite a few of these. These are just some of the more common exotoxins. This is not all of them by any means at all. And there's a little bit more information here that goes into a little more detail about these different types of toxins, and I'll kind of let you read through that on your own. In the next video, we'll get into the rest of these virulence factors that can be produced by bacteria and viral pathogens.